All right, let's get into it. Every single time a cell divides, it has to pull off something incredible. It's like the perfect crime. It copies its entire genetic blueprint. We're talking billions of letters, with almost zero errors. So today, we're going to treat this whole process like a molecular whodunit. We are on the case of the copied code, and we're going to follow the clues that finally unraveled the mystery of DNA replication.
Our investigation, well, it kicks off right here, way back in 1953. When Watson and Crick unveiled that beautiful double helix, they dropped this little bombshell of a clue in their paper. They just knew the structure itself was hinting at how it could be copied, but the exact method? That was still a total mystery. You could say this one sentence officially opened the case.
So here's our case file for today. First, we'll look at the initial coded message. Then, we'll line up the usual suspects, the three big theories that were floating around. After that, we dive into the one experiment that totally cracked the case. From there, we'll investigate the machinery, the enzymes that do all the heavy lifting, compare how they operate in different jurisdictions, and then we'll wrap it all up with a final review.
Okay, so that double helix discovery, that was the first major break in the case. The whole idea that A always pairs with T and G always pairs with C was a huge clue. It basically shouted that one strand could be a perfect template for making the other. But how? How did the cell actually use that template? Did it make a copy of the whole thing and leave the original alone? Or did it pull the original apart to do the job? The mystery was a long way from being solved.
And that leads us to the next phase of our investigation, rounding up the primary suspects. In the years after Watson and Crick, the scientific community cooked up three main theories, three competing models for how the cell could be pulling off this massive replication heist.
Let's check out their MOs, shall we? First up, we have the conservative model. Its whole method was keep the original parent DNA totally intact and use it as a mold to make a brand new daughter molecule. Suspect number 2 was the semi-conservative model. This one suggested the parent DNA would unwind and then each of its two strands would become a template for a new one. And last, we have the dispersive model. This one was kind of chaotic. It proposed that the parent molecule gets chopped up into little bits and the new DNA is just synthesized in between all the old fragments. As you can see, each suspect predicted a totally different outcome.
To solve a case this big, we needed some brilliant detectives, and that's where Matthew Meltzelson and Franklin Stahl come in. In 1958, they put together this incredibly elegant experiment. It's so clever that people call it the most beautiful experiment in biology. And it was designed for one thing, to finger the one true culprit.
Now this is where the genius of their plan really shines. They used two different types of nitrogen, a heavy version, N15, and the normal, light version, N14. First, they grew bacteria in the heavy nitrogen, so all of its DNA became heavy. Then, they switched it to a light nitrogen medium. That meant any new DNA the bacteria made would have to be light. This was basically their version of dusting for fingerprints. It let them track the original parent DNA versus the new daughter DNA. Pretty clever, right?
And boom! Here's the first break in the case. After one generation of growth, they took the DNA and spun it in a centrifuge. Now, the conservative model predicted they'd see two separate bands, one heavy, one light. But that's not what they found. They saw only a single band, right in the middle, at an intermediate density. This clue was huge. It immediately eliminated the conservative model from the suspect list. It just couldn't explain this hybrid DNA.
But the real smoking gun, the final, irrefutable evidence, came after round 2. This time, when they spun the DNA, two distinct bands showed up. One was that same hybrid DNA from the first generation, but the other was a brand new band made entirely of light nitrogen. This result, it was a perfect match for what the semi-conservative model predicted. And it also ruled out the dispersive model, which would have just given them a single band that got progressively lighter over time. Case closed. The culprit was, without a doubt, semi-conservative replication.
Okay, but knowing who did it is only half the story. Now we got to figure out how they did it. We're moving on from the mastermind behind the operation to the crew on the ground. We're going to investigate the molecular machinery, this whole gang of enzymes that are responsible for carrying out this incredibly sophisticated job.
All the action goes down right here, at the scene of the crime, the replication fork. You can picture it as this Y-shaped junction where the DNA double helix is actively being unzipped. This exposes the two parent strands so they can be used as templates. It's not a static place, it's a dynamic, moving worksite.
Now, here's where a little complication pops up for our crew. The two DNA strands are anti-parallel.
They run in opposite directions. But the main enzyme, DNA polymerase, it's a one-way worker. It can only build in the 5' to 3' direction. So what's the cell's clever solution? It synthesizes the two new strands differently. One strand, the leading strand, gets made in one nice continuous piece, but the other one, the lagging strand, has to be made backwards, in little short bursts that get stitched together later.
And the solution is actually pretty ingenious. The lagging strand gets built in these little pieces called Okazaki fragments, named for the scientist who found them. These fragments are really the key evidence that proves replication on this strand has to be discontinuous. Each little fragment is made in the correct 5' to 3' direction, and then, later on, the cell just stitches them all together to make a complete strand.
So, who are the key players in this enzymatic gang? Let's meet the toolkit. You've got helicase, that's the specialist that unwinds and unzips the DNA. Then there's primase, which lays down a little RNA primer. Think of it like a starting block for the main builder. Ligase is the master welder, coming in to seal up the gaps between those Okazaki fragments. And topoisomerase, it works ahead of the whole operation, relieving all the tension and stress that builds up from unwinding the DNA.
And of course, we can't forget the boss of the whole operation, DNA polymerase. In prokaryotes, you've got a few of them, but the two main players in replication are Pol3 and Pol1. Think of DNA polymerase III as the main workhorse. It's the master builder doing most of the synthesis on both strands. DNA polymerase I is more like the cleanup crew. It comes in after the fact to remove those RNA primers and fill in the remaining gaps.
Now, it's important to know that this operation doesn't look exactly the same everywhere. The basic mechanism, the whole semi-conservative thing, that's universal. But the logistics, they change when we move between different jurisdictions. We're talking from the simple circular chromosomes of prokaryotes to the big, complex, linear chromosomes of eukaryotes. So really, the key difference is the sheer scale of the operation. Prokaryotes, they have these small circular genomes, and they usually just need one starting point, one origin of replication. But eukaryotes, with their massive linear genomes, they have to be way more efficient. They use thousands of origins all at the same time, creating these multiple replication bubbles that just expand and merge together. Honestly, it's the only way they could ever hope to copy that enormous amount of DNA in a reasonable amount of time.
All right, we've gathered all the evidence, we've identified the culprits and their methods. Now it's time to consolidate our notes for the final exam. Let's run through a quick review of the absolute must-know concepts from our investigation.
Here's your final checklist for the case file. You've got to be able to explain each of these. Semi-conservative replication, the winning model. The incredibly elegant Maselson-Stahl experiment that proved it. The structure of the replication fork. The difference between leading and lagging strand synthesis. Why Okazaki fragments are so necessary. And of course, the jobs of the key enzymes, like helicase, the main builder DNA polymerase III, and the sealer, ligase. Let's test your memory. Rapid-fire question number 1. Which of those three models was immediately thrown out after that first generation of bacteria gave them that single hybrid band of DNA?
Question 2. What's the specific job of DNA ligase? What's that one crucial step it performs, especially on the lagging strand?
And last one, question 3. Thinking about those different jurisdictions, what is the single biggest difference in strategy between prokaryotes and eukaryotes when it comes to initiating replication?
We're going to end on this thought. The case of the copied code may be closed, but the implications are just massive. The cell copies its entire genome, an encyclopedia of information with fewer mistakes than a professional typist makes on a single page, and it does it all in just a few hours. So the final question to think about is this. What does this unbelievable, high-stakes perfection tell us about the absolute, non-negotiable demands that life itself requires to exist?